Start. Hello everyone, I'm Jason Lee.
I'm Oscar.
And I'm Alex Polvada, and we did our project on how can we use microscopy to better understand the plaque buildup in brains of Alzheimer's patients. So we're going to use fluorescence microscopy to locate where the plaque builds up in the brain, and then we're going to use electron microscopy to examine the structure of the protein of beta amyloid. The relevance of this is Alzheimer's is one of the most predominant senile diseases, and by analyzing how the 3D structure of the protein looks, we could possibly develop a cure. From the data, we hope to understand why the plaques build up and where they build up, and then create a cure that can eliminate Alzheimer's.
So some of the social ramifications of research into Alzheimer's is that those affected by the disease tend to lose their memories and then eventually even their identities. And is it mic? Okay. And this especially affects the families of those who are affected by Alzheimer's because the affected basically no longer recognize their family, and also constant care is required toward the later stages. And also there was a 71% increase in deaths to Alzheimer's in the U.S. from the year 2000 to 2013. And the number of seniors in the U.S. projected to have Alzheimer's by the year 2025 is going to increase from 14% to as much as 60% in certain states.
So as an overview of the equipment that we're going to use for our research, we have fluorescence optic microscopy on the left. It functions by exciting an electron which emits a photon, a light particle, and back into the eyepiece. That's how you see the image. And we have the electron microscope on the right, which features electron beam shooting through the sample onto the fluorescent screen. And what you see, the image that you see, is basically a electron density image of the through the sample. So now Jason's gonna elaborate into how we're gonna specifically use those equipment to foster our research.
So first of all, let me talk about the particular form of fluorescent microscopy. We propose to use a method called Sted Microscopy, which basically the premise of fluorescence microscopy is that a laser is shown onto the sample which has been prepared with fluorescent markers. This laser contains photons which excite those markers, and those markers in turn produce their own photons, which are then captured by a detector. With Sted microscopy, a secondary laser in the shape of a ring, as you can see in that middle picture on the bottom row, is sort of wrapped around the main excitation laser. This causes that main laser to be reduced to a much narrower diameter, which can dramatically increase the resolution of the image, enable us to image in far greater detail and at far smaller sizes. So how this optical fluorescent microscopy will be applied is that we will use primarily antibody quantum dot affinity pairs in order to mark our specimens, as our specimens are going to be primarily protein-based neurotic plaques in diseased brain tissue. The antibodies bind to specific proteins, and the quantum dots, which are made of nanocrystal heavy metals, shine very brightly when excited by the laser, which can enable us to have a much brighter contrast in our image. This fluorescence microscopy can help us compare known healthy brain tissue with our samples, which can confirm the presence of beta amyloid plaques. The electron microscopy phase can enable us to directly image and analyze the structures of those proteins, of misfolded proteins inside the neurotic plaques, which can enable us to learn a lot more about how these, what exactly went wrong in the formation of these proteins and what can be done to stop it. So some of our primary challenges that we will face is finding samples that will correctly match the conditions inside the brains of the humans with Alzheimer's. One of the ways we can get around this is by using brain tissue obtained via autopsy, but the main drawback to that is that the brain tissue could be potentially degraded by storage and chemicals.
So for the literary review, we're gonna run over the foundation's published papers that serve as the foundation of our research. So as you can see on the chart, since we're primarily interested in plaques, so it's colored in green. We have different mice species in columns, And as you can figure out, they fall pretty uniformly and consistently between the time period of 9 months to 12 months. That's how we figured out our test subject and our the specific time frame that we want to work with.
Some other research that we did was we used articles to find out that the beta amyloid protein was the one that was causing the plaque buildup in the brain, and which is the one that blocks the synapses from firing and causes Alzheimer's. Another thing that we found was most of these articles did not deal with the actual structure of the protein. Rather, they analyzed the effects, so that's why we're interested in getting a 3D image so we could learn about the proteins and see how they fold so we could develop a cure. Okay. Future direction. We hope to use the electron microscopy to make a 3D model of the beta amyloid, understand it, see how it works, and then hopefully develop a cure and test different medicines to see which ones work best to ultimately end Alzheimer's.
So here is a list of the articles that we have referenced in order to compile our PowerPoint. And
Thank you.
thank you all for listening.
Very interesting, first of all. And what I like is you're presenting two different approaches to the same project, which is really interesting, right? Fluorescent microscopy and electron microscopy. So since you are doing so, I'm guessing that you are expecting to extract different information from the two techniques. So what I would like to ask you is, what kind of information do you expect to get from fluorescent microscopy approach, and what would be the different information you expect to get from electron microscopy?
For the fluorescence, we're wondering mainly where the plaque builds up and seeing how, where and why it builds up, and also just to analyze the difference between the healthy beta amyloid and the unhealthy beta amyloid, so then we could extract and identify the unhealthy ones, which we could then put in the electron microscope and analyze the unhealthy proteins to see how they form and really see how they react to different medicines and see if we could reverse Alzheimer's.
So on your future direction slide, or your conclusion slide, you said that you would identify the structure and then we could start to look for a cure. So after you do have the structure, then what would be the next step that researchers would take?
Well, we could try to analyze the protein, see how they bind, and see, maybe get an idea of what medicines we could use to start reversing it.
So you have seen the structure between a healthy and a defective one, right?
Yes.
What's the, can you kind of tell us the difference, or?
Well, unhealthy amyloid beta proteins tend to cluster in a form of a plaque in the brain. So that's the primary reason why we want to study the locations. Once again, the location, their locations, and then further on move on to study their interior structures to develop a cure, as aforementioned by my other fellows.
So unhealthy one tends to like cluster together and stuff.
Yes, and block the neuron transmission process.
Other questions? I saw another hand raised over here. Yes.
So you said you're going to be testing on mice 9 months, 12 months, right? And so why did you choose to test on them? Like what similarities do they have to the human brain that you're hoping to exploit? Like, do mice also contract Alzheimer's as well, or why are you looking?
Yes, they contract Alzheimer's, and it's more of like a moral dilemma, because it's kind of, if we try to use an actual patient, then that would be unethical. And then if we try to use the autopsy one, then the chemicals can interfere. So right now, the mice seems like the best solution, but we are going to try to maybe use the brains of autopsies of deceased patients, so we could get like a 2-scope to see how one is like right away without chemical treating with the mouse, and then one with the human with chemical treating, to get a more comprehensive idea of how the proteins work.
One more question?
got one more about the structure. Now, in the future, are you guys looking into like destroying that, or to rebuild it, or something like renovating it? You know what I'm saying? Try to fix the structure, or just completely destroy it.
One of the more immediate fixes we predict could be done to dissipate these plaques is sort of develop a different compound that can prevent those beta amyloid proteins from sticking together in the first place. And, sort of a compound that binds to the beta amyloid prevents them from binding to each other in turn. With greater knowledge of the structures, we can maybe go a step further and identify the cause of why those beta amyloid proteins misfold and misform in the first place, which would prevent any sort of plaques from forming and solve a great problem in this research.